Hello, my name is David Guevara and I am a graduate student at Utah State University. Today, I'm going to talk about prediction and classification of virus families, subfamilies, and human MERS, SARS-CoV, and SARS-CoV-2 protein-protein interactions prediction using machine learning. Proteins usually need to interact with other proteins to carry out their functions. This is a protein-protein interaction. Having knowledge about these interactions is important to discover new drug targets, enhancing or disrupting these interactions so that diseases can be treated. We can find new interactions by carrying out experiments and finding them in the laboratory. This can be done with high or low throughput methods. High throughput methods are unreliable and low throughput methods are costly and give a low amount of data. This is why predicting these interactions with machine learning can be useful. For our experiments, we tested random forests and neural networks. We developed a 4-phase prediction and classification system where the user inputs a viral and a human protein. The system goes through all phases, classifying the virus protein and finally predicting whether both proteins interact or not. Before running through the prediction models, it verifies the length of both amino acid sequences. If one or both are shorter than 31, it stops the prediction. And if it passes, it encodes both proteins in a way that all sequences have the same length of data. The first phase identifies whether the viral protein is actually a viral protein or not, and if it is, it tries to predict if it belongs to the ssRNA negative class. The second phase identifies whether the virus is a coronavirus, or more specifically, if it belongs to the Coronaviridae family. The third phase identifies whether the protein belongs to a MERS or SARS virus. It is pretty difficult to distinguish between SARS-1 and SARS-2, so we decided to group both in one class after multiple tries. Finally, the fourth phase predicts if there is an interaction between both proteins. There is a different model for each one of the three viruses. If the phase 3 predicts it is a MERS protein, it runs through the human MERS model. If it predicts it is a SARS protein, it runs through both the human SARS-1 and SARS-2 models. As for the data collecting, for phase 1, we gathered data from NCBI, Viper, and Genome.jp. For phases 2 and 3, we gathered data just from NCBI. For phase 4, we looked for papers that had experimentally proven interactions. Then, we complemented the list with data from UniProt, Intact, and BioGrid. All duplicate interactions were removed. For phase 4, we had to create a negative dataset. The most usual way to create datasets is randomly pairing proteins that have not been experimentally proven, and then filtered by their subcellular locations. For our experiments, we found another way of creating the dataset, which was crossing all virus proteins against human proteins, rank them by the similarity score, and picking the interactions with lowest score. As for data cleaning, for phase 3, we found out many sequences had a lot of X amino acids. So we removed the sequences with 2 or more consecutive X's, and for phases 1 to 3, we used CityHit to remove sequences with high similarity. For data encoding, we used iFeature, which is a Python package that, among its tools, it can encode amino acid sequences into up to 38 feature descriptors, even though almost half of them need all sequences to be the same length, which is not the case here. Neural networks were built using Keras, the TensorFlow high-level API. The neural networks consist of a pair of one-dimensional convolutional layers, followed by max pooling, applying batch normalization on the second layer output, and finally 2 dense or linear layers, with a dropout layer between them. We trained the models reducing the amount of data points of all classes, so they all had the same amount of data, using 10% of that data to test, and also adding the surplus data from the reduced classes to the test set. For now, we have tested the models with dipeptide composition encoding, or DPC. We selected the best models according to the MCC, or Matthews Correlation Coefficient, because it is the single score that best describes the results, according to some claims, because it takes into account every value in the confusion matrix. The results show that the neural networks is performing better than the random forests in all phases, especially in phase 1. It is interesting to note that phase 1, the one that distinguishes between non-virus, non-ssRNA negative virus, and ssRNA negative virus, is the one with the lowest score. It may be due to the fact that among the NCBI sequences there were lots of sequences with just fragments of the proteins, so it may be adding noise there. This won't be a problem with predicting interactions though, because the ssRNA negative it's getting 97% accuracy. And also, if the user is sure about the protein belonging to an SSRNA negative virus, or a coronavirus for that matter, they can choose to skip any phase. Looking at these results, we want to further improve the network performance in phases 1 and 4, so it's more reliable. To do that, we'll try with more hyperparameters and also other encodings like AAC or QS order, and combinations between them like joining DPC with QS order. This may help raising up the score. We will also develop the web server so it is available for everyone who would like to use our software. It would also be interesting to complement the tool with more virus options besides SARS and MERS, but that also relies on data availability. Thank you for joining our presentation. If you want to contact us, you can go to bioinfo or bioinfocore.usu.edu or by email to david.guevara@usu.edu and rcandle@usu.edu.